thank you all for attending today. I'm really excited to share some of the work that we've been doing with everyone. Today, I'll focus on a few main areas. First, I'll tell you a little bit about who we are as a research team and the type of work that we do, in particular, how we use infant brain imaging to study brain development in autism. I'll talk a little bit about what we've discovered in terms of early brain changes in autism and how we might be able to use brain scans to predict later diagnoses in certain children. And I'll talk a little bit about how some of our findings over the past few years have started to point to differences, in particular in the development of the visual system during infancy. And I'll end this webinar telling you a little bit about what we're doing now and talking about what we call our early prediction study. So our team is we call ourselves the Infant Brain Imaging Study, or Ibis Network, and we, as a team and as a study, are the first and largest longitudinal study of brain development during infancy and autism. We have been working as a team for over 15 years to study how autism develops, and in particular, how the brain develops in autism. Our initial cohort involved the study of younger siblings of children with autism and included 400 participating infants and their families. And those infants first joined the study in the early 2000s and are now entering adolescence. And we've continued to follow those kids so that we can learn about how autism develops in terms of brain and behavior over this sort of period spanning, infancy all the way through adolescence and hopefully into young adulthood. We are also currently collecting a new cohort of infants, and this is our Ibis Early Prediction Study, or I might refer to it as Ibis EP throughout this presentation. And that new cohort of infants is designed to help us replicate some of the original findings of the study, and also to think in particular about how we can use brain imaging data to help us predict later areas of concern for children and or later diagnoses. Our work focuses on what we call the infant sibling study design, and this capitalizes on the high heritability that's observed in autism. So there's a large genetic component to autism, and it runs in families. Younger siblings of children with autism can be followed prospectively to study the emergence or the development of autism. And this is because about 1 in 5 of those younger siblings will develop autism over the course of their childhood, which puts them at about 10 times higher likelihood for receiving a diagnosis than children in the general population. So we use this study design so that we can collect enough data on enough infants who go on to develop autism and make meaning or understand more about their brain development. Our study timeline is shown here. So we recruit families who have an older sibling with autism, and I might refer to them throughout the talk today as the proband. And these families have at least one older child with autism. Sometimes they have more than one. And they have a new infant born into the family. So these infants are followed for their first assessment around 6 months of age, and again around their first and second birthday. And at each of these visits, we collect both MRI and behavioral data. And the MRI is what really, in many ways, sets this work apart from a lot of other studies out there that have been following infant siblings for quite some time. And our study uses MRI, or magnetic resonance imaging, as a window into the developing brain. So MRI is safe for repeated use. There's no radiation, and it can be done during natural sleep. So what you see here in these pictures it's a mother and child who participated in our study. And on the left-hand side, you can see that she's sitting in the scanning room, the scanning environment next to the MRI scanner, and helping her child fall asleep. So she's rocking her child. Once the baby is asleep, she then helps position her child on the bed of the MRI, and later we'll be pushing the infant into the scanner to do the scan. In addition to the parents who help make our work possible, we also have what we refer to lovingly among our team and staff as our baby whisperers. So these are the highly trained staff members of our team who actually go into the bore of scanner with the infant to make sure that they stay awake and comfortable during this process. The scan takes about an hour to complete, and we have a pretty high success rate, about 90% success rate for these scans. So once an infant has fallen asleep and gone into the scanner, 90% of the time we have them, they stay asleep the entire time and we get the images that we need. During this 1 hour protocol, we collect different types of images that help us to understand both the structure and the function of the brain. And so in the example image that I show here kind of at the bottom is of an infant who's actually fell asleep sucking their thumb in the scanner. So you can see that their hand is up by their face. And you can see their eyes in the image and their skull and their brain. And in this particular image, it's called a structural MRI image. What we look at is differences in the sort of color or contrast in the images that help us define different parts and components of the brain. So what's shown here in this sort of like lighter color is the white matter of the brain. And white matter comprises long range axonal connections between different parts of the brain. And then you can see sort of the folder outed cortex of the brain. And this is the brain's outermost layer of cells that contains most of the brain's neurons. And we use these types of images to quantify how brain development happens across the first 2 years of life. And through this type of approach, I think I would say we've made two key discoveries in this work. The first is that brain changes are apparent in autism during infancy, and they precede changes in most behaviors. And I'll talk a little bit about that timeline in the next slide. And I think the other key discovery we have made is that we can use machine learning to predict autism diagnoses from brain scans that we take in the first year of life. So we can identify infants with pretty high accuracy who will go on to receive diagnoses of autism more than a year later. And I just want to highlight a few of the differences in the brain that we've observed so far in these infants who develop autism. So just to orient you a little bit, at the bottom of the image here, you can see a timeline, and this spans from 6 months to 36 months or 3 years of age, and sort of breaks down this timeline into what we think of as the autism prodrome and then the emergence of symptoms. So the prodrome is sort of lasting anywhere from 6 months to about 18 months, where most children are sort of not showing, it's certainly not showing overt signs of autism, but maybe also not even really showing many some more subtle changes in behavior. But sometime around, between the first birthday and the second year of life, we start to see this emergence of symptoms that persist thereafter. And sort of along the same timeline, we have various differences in how the brain is developing. So if we look here at the top of this figure, what I'm going to show first is what we have found in terms of increased extraaxial cerebrospinal fluid volumes. So cerebrospinal fluid is the fluid that surrounds the outer surface of your brain. It's colored red here in this image. And everybody has CSF around the outside of their brain, and we are only really over the last, I would say, several years learning what what the purpose of CSF does and what its function is in the brain, but we're starting to understand that it's an important part of a waste clearance system. So it removes sort of byproducts of the chemical and neuronal reactions that happen in the brain, and it filters those out through what's called the glymphatic system. And you can kind of think of it as sort of like how your body filters blood through the kidneys. It's a similar type of concept. But what we're learning in autism is that starting at the first time point that we measure the brain, which is at 6 months of age, we're seeing that infants who go on to develop autism have an increased volume of the CSF surrounding the brain, and that increase remains through the period of 3 years of age. And what this may mean in terms of mechanism is still under investigation by our team, but it suggests that there may be differences in the way in which the brain is clearing waste products. We also see atypical patterns of white matter maturation or development. And as I kind of mentioned before, when I was showing you the MRI image of the infant, the white matter, you can think of these as like the information highways in the brain. They carry information between different parts of the brain that's responsible for processing, various levels of information. So, for example, white matter connections will carry information between parts of the brain that are responsible for receiving visual information from the eyes to other parts of the brain that help us make meaning of what that visual information is. And what we observe in autism is that as early as we've measured it at 6 months of age, there's a difference in how the white matter looks in children with autism, and that this difference sort of continues to persist, but over time. And it's really more developmental trajectory that's different in autism than in typically developing children. So it's not a particular sort of mechanistic difference. It really just seems that it matures at a different rate. We also observe that the amygdala overgrows in autism, and this actually happens developmentally across the latter part of that first year of life. So at 6 months of age, children who later develop autism don't show any differences in the volume of their amygdala, but sometime between 6 months and 12 months, they have this period of large growth where they ultimately end up with larger volumes of the amygdala. And the amygdala is a part of the brain that's important for various aspects of brain function. And one of those includes sort of assessing threat response. It's often implicated in anxiety. And is also responsible for sort of orienting us to different cues in the environment and reading their salience. So it has a broad sort of implication across various behaviors. At the same time that the amygdala is overgrowing, we see the overall sort of outer surface of the brain, or the cortex, also overgrowing. And this happens again. It's not present at 6 months of age, but occurs sometime between 6 and 12 months, where we ultimately end up with like a larger surface, a larger cortical surface of the brain. And then finally, we see an overgrowth of the overall size of the brain. And this starts to happen between the first and second birthday. So again, infants are not born with enlarged brains in our cohort, but they develop them over time. And the mechanisms that might be at play for these various types of overgrowth in the amygdala, in the cortical surface, and in the overall brain are still under investigation, but it could arise through a variety of mechanisms, one of which is that there's more neurons in the brain and they're creating more connections. And as those connections sort of mature, they grow in size, the brain grows in size. Alternatively, across the same developmental period, in typical development, the brain prunes itself and circuits are pruned to become more efficient. And it may also be that these circuits are not pruning over time. So I would say this is almost 2 decades worth of work that I've summarized in one slide. So I know that's a lot of information, but I think what I really want you to take away from this is that there's no single area in the brain that's different in autism. There's no single component of brain development that's different in autism. It's very much a broad difference in brain development that we're observing here. But the piece of this that I think is so fascinating is that many of these changes are developmental in nature, meaning they sort of come online over time. And I think that suggests to us that there are mechanisms or times in development that we might be able to target our intervention, before some of these things have started to change or very early on in that process. And it's likely to help us ultimately have the best positive impact on brain and behavior development in children. And so after, sort of establishing a base of how does brain development happen in autism, we became very interested in whether we could use this information about brain development and from brain scans to help us think about who's going to go on to develop autism. And we have 2 main studies that worked on this particular approach. And one of those looked primarily at features of the cortical surface. So this is the part of the brain, the outer part of the brain containing most of the brain's neurons, and it's growing too quickly or more quickly in autism than typically developing infants in the second half of the first year of life. And we looked at features of the size of the cortex across different regions of the brain. And we also looked at the thickness of the cortex across different regions of the brain. And we fed this information into machine learning models models that were then trained in a subset of the data to sort of predict or learn what aspects of the brain are related to a diagnosis of autism versus, controls. And then we showed that trained algorithm novel new data from different participants and said, okay, now that you've learned how these relationships work between brain and diagnostic outcome, what do you predict the outcome is for these infants? And this study resulted in what we refer to as a positive predictive value of 80%. And a positive predictive value just means out of all of the results that the algorithm returned that said autism, 80% of them were actually autism cases. And in our sample that went into this particular algorithm, 34 infants received a diagnosis of autism in the study, and the algorithm correctly predicted 30 of those 34 as being in the autism group based on brain imaging scans that were taken a year or more than a year before that diagnosis was made. We then used a very similar approach, but using a different type of imaging data. And in this case, we use what's called functional connectivity. And functional connectivity measures the degree to which two brain regions are on and activated at the same time. And we looked at the patterns of connectivity across the whole brain. So how is each region connected to all the other regions in the brain? And we put that information into an algorithm, same type of process that I described before. And in this case, the positive predictive value for the autism diagnosis was 100%, meaning that if the algorithm said the infant was fell in the autism group, then they in fact did have an autism diagnosis. And in this case, this algorithm picked up 9 out of 11 total infants in the sample that were diagnosed with autism. So these studies were really interesting and in many ways prompted a lot of interest from our group in thinking about how we could move this forward in some way that was clinically meaningful. So I'll say first that these results are quite striking in the sense that this MRI data and prediction outperforms behavioral prediction from screening tools that are used regularly in clinical settings, meaning that it was more accurate and was more likely to be correct. This was the first evidence that MRI could be used potentially as a screening tool, or in particular, a second level screening tool in the context of these families who are already at high likelihood. But these are relatively small studies, and machine learning algorithms are they're likely to sort of be biased. So it's important for us to show that we can replicate these findings in a new cohort and in a new sample of data and show that they're robust and that they're something that we could take forward to use kind of in a future, if not clinical sense, at least a research sense to think about designing and implementing interventions. And this is the goal in many ways of our new cohort and data collection that I'll tell you a little bit more about at the end of this. And so now I'll switch a little bit to talk about the visual system and how many of the findings that we reported before kind of start to center around the visual system. So the visual system is the sort of system in the brain that's responsible for processing visual information, interpreting what this information is, and also in many ways guiding what we actually look at in the environment. So visual information is passed through the retina to a relay center in the thalamus in the brain, and then to the primary visual cortex. From there, it's processed to other parts of the brain that are responsible for sort of identifying what is it that we're looking at, is it moving or is it still, and making kind of categorizations about the visual information. That's then relayed to other parts of the brain that are responsible for decision making and orienting of attention, for example. So the visual system in many ways is sort of like the ground zero for how we get at one major sensory modality, which is visual input, into the brain and how we react to that. Understanding visual system development in autism may help us understand many of the early differences in behavior that are observed in these infants, and in particular, differences in visual attention and where infants are looking in the environment. And I'll talk a little bit more about that and the evidence we have for that later. So, what's different about visual system development in autism? Um, so, one of the main things that we've noted in our studies so far is that, I told you the overall size of the cortex hyperexpands from 6 to 12 months in autism. But if we take a look regionally across different parts of the brain, the areas with the largest expansion or the fastest rate of expansion are actually areas that are within the visual system or directly connected to the visual system. So these are sort of parts of the brain that are kind of in the occipital lobe and occipital and temporal lobes. And it's many of these same regions that are also the top contributors to our prediction algorithm. So if you'll recall, I told you that we used cortical surface features across the brain as one of the predictors of autism diagnosis. And if you kind of look under the hood in those machine learning algorithms, what you see is that these hyper expanding visual areas are also contributing to the prediction of autism. And this could be related again to a variety of different underlying mechanisms, but it's sort of our first clue that there may be something atypical about how visual system, at least to the structure of the visual system, develops in these infants. We then, as I mentioned, we have a study showing that the amygdala starts to overgrow around the same period of time. And the amygdala is, among many things, responsible for orienting visual attention to salient or important aspects of the visual environment. The amygdala shares direct anatomical connections to the visual cortex. And so the timing of these two areas, sort of hyper-expanding and overgrowing, was very interesting to us, the fact that we see these things kind of happening at the same time in development. And we also have shown in the past, and in this paper, that the rate of amygdala growth is related to differences in social behavior. And in particular, the faster that amygdala grows, the more likely that child will go on to have greater impairments in social behaviors. So we're starting to see how aspects of the visual system, or at least parts of the brain that are connected to primary visual system, are developing differently across that latter part of the first year of life. We then followed up that study to ask about the functional connectivity of the amygdala. So it's growing fast from 6 to 12 months, but what are the implications of that in terms of how this area talks to other parts of the brain? And what we've found, and this paper is actually going to be published next month, what we ended up finding was that we looked at amygdala connectivity across the whole brain. And the one aspect of connectivity that was different in children who had a family history of autism, so these were all of those high likelihood infants, was that the connection between the amygdala and the visual cortex was weaker. So we found in this case a functional and perhaps consequence of that overgrowth. So maybe these regions are overgrowing at the same time. It's possible that they have more structural connections, that we haven't measured that, but they have weaker. They're not in tune with each other. So there's definitely a difference in the way that these two regions are communicating with each other in these infants with a genetic predisposition to autism. And the weaker that connectivity was between those two regions, the amygdala and the visual cortex, the lower those infants' motor and communication abilities were. So here we start to see, sort of a functional behavioral consequence of a difference in connectivity between two aspects of the visual system. And interestingly, these are not, core features of autism, but they're early emerging differences. So we know that infants who later develop autism often show motor delays and communication delays that are detectable by the first birthday. And we've also shown that there are differences in aspects of visual attention and how these are related to aspects of visual system structural connectivity. So Visual orienting is delayed in autism, and what visual orienting is, which you can kind of see an image of here. So how we measure this is we have infants come in and they do an eye tracking task. So they're looking at a screen and there's a camera above the screen that captures where their pupil is looking on the screen. And this is a really common way to measure behavior in young infants who can't speak, because we interpret that where their eyes are looking is where they're attending. So at 6 months of age, we show infants this type of orienting paradigm. And what happens here is initially there's a central stimulus, which in this case is a green dot. And after a few seconds, a peripheral stimulus comes up, and this is a green dot with the sort of black circle around it. And we measure how long it takes the infant to disengage from looking at the central target and to look at the peripheral target. And what we end up seeing is that infants with autism are slower to do that. So they're sort of less quick at picking up on the peripheral stimulus, or at least less interested or looking at it immediately. So there's a difference there in how long it takes for them to look. And the latency, so how long it takes for them to look at that additional stimulus, is related to the development of white matter in the brain, in a particular tract that connects the left and right visual cortical areas. So, this is suggesting to us, perhaps this is, to my knowledge, is some of the first evidence that there is a sort of a link between visual orienting attentional behavior and brain structure within the visual system that is different in autism. So if you look at this graph here, this is just a pictorialization of the data, but what we ended up finding was that there's a difference in how that orienting latency is related to white matter in the brain between autism and control. So it really starts to suggest that the ways in which these behaviors are encoded in the brain or embedded in the brain may be very different between these two groups of infants. In another study of ours, we've also shown that aspects of initiation of joint attention are related to the development of visual system and its connections with other brain areas. So a joint attention is the initiation of joint attention is the process by which, in this case, a child engages with another person, and an adult in this image here, to orient their attention away from something to whatever they want to share interest in. So in this picture here, mom was originally looking at this green balloon, but the child wants her to look at this pink kind of beach ball, and she points to get her mom to look at that. And this type of behavior, this initiation of joint attention, is something that we know is different in autism, and it develops kind of early on. We also know that it's a very important behavior for learning, and in particular for language learning, and other aspects of social learning. And it's sort of a central ingredient for that learning process. And this study showed that the rate at which children do this, the more often that they orient the attention of another person to what they're looking at or want to engage in, the stronger the connectivity within their visual networks. So if we start to kind of put all of this together into a developmental timeline, I think it helps us see how some of these things may unfold and the particular role that visual system may have. So we've shown that at 6 months, there are links between orienting behavior and the structure of connections between the left and right visual areas in the brain. At 12 months of age, we're showing that the ways in which children orient joint attention between themselves and another person is related to connectivity within the visual system. We know that the sort of structural cortical areas that process visual information grow more quickly in autism, or they hyperexpand across this period of time. It's the same period of time we see the amygdala overgrowing. And we know that amygdala overgrowth is related to later social problems. And if we look at the end of this sort of overgrowth period for the amygdala and the visual cortex, at the end of this period, they end up being less functionally connected. So they're not really in tune with each other in the same way. And I'll give you a teaser to say that these things are not specific to the infant period, but they emerge here and they persist. So we have data in the same infants that we followed up between 7 and 11 years of age. And what we see here is that we have a weakened connectivity between the visual networks here at the back of the brain and the salience network. And the salience network is thought to be responsible for helping sort of orient people to salient information or important information in the environment. And what we see is the connectivity between these two networks is weakened in children who have social impairments at 7 to 11 years of age. And that this is actually not specific to autism diagnosis in particular, but also to high likelihood infants who have problems with social behavior, but may not quite meet the diagnostic threshold. And so this really suggests to us that there's, a sort of heterogeneous neural component to social behavior that's embedded in many ways, perhaps in visual system function and its relationships with other parts of the brain. And I was particularly interested because we have this familial sample and understanding how we might be able to probe whether there are any components of these differences in the visual system that are in fact sort of traceable to genetic factors or heritable factors. In this case, we used familial autistic traits to get at this because studies of molecular genetics, so as many of you may know, if you participate in SPARK, requires a many thousands of people to sort of study genetic variation because of just the sheer size of the genome and the number of people we need to really search that. So instead of taking a molecular genetics approach to looking at this, we decided to take an approach where we look at a heritable phenotype that travels in families. And in this case, we looked at autistic traits in the older diagnosed sibling in the family, so the proband. And we have these available to us at the time at which the infant sibling enters the study. So it's also a nice sort of predictive tool if it works in that way. So what we did in the study was we said, okay, we're going to look at the autistic trait load within that older proband, varying from kind of low levels of autistic traits to higher levels. And what I mean here by traits is sort of the endorsement of more autistic behaviors by a caregiver. So this is all caregiver report. And what we ended up finding in this paper, which was pretty interesting to us, was that 3 out of 4 infants within our sample who went on to develop autism had probands with higher than average levels of autistic traits as defined by our sample. And so if your proband had higher than average levels of traits, you were about 3 times more likely than other infants within our high likelihood sample to develop autism. And so we use that same measure of autistic traits, and we then related it to aspects of brain development in the younger sibling. And we said, okay, if we know that these level of autistic traits in the older child is predictive of the younger child's diagnosis, might it also be sort of predictive of a genetic liability or genetic predisposition, and how does that play out in the brain? And what we ended up finding was that higher levels of autistic traits in probands related to weaker functional connectivity in visual networks, larger cortical surface area in visual areas, and atypical white matter in visual tracts. So all the areas that we'd already shown develop differently in autism do so as in sort of a graded way based on the genetic liability running within the family. And obviously this is sort of an interpretation of how the familial traits operate. And we need to kind of look at this more closely. But I think this suggests to us that looking within brain areas in the visual system for molecular genetic studies that are focused might be a really interesting next step. And in fact, there was a paper that came out just about a year before we did this work that suggested that using postmortem data, that there was robust differential gene expression in the visual cortex in individuals with autism above and beyond other parts of the brain. So it's very much kind of lining up with this idea that there may be a genetic predisposition to atypical development of the visual system in autism, and that this may ultimately have sort of cascading effects on development. So how does this fit with what we know about behavioral features in autism and sort of what might this mean more broadly for how we think about autism and the development of autism? So we know, and there have been many studies that have replicated this type of finding, that the viewing of social information using these eye tracking studies are different in autism, and that this occurs very early in development during infancy, but it really persists. Across the lifespan in individuals with autism, and that on average, individuals diagnosed with autism spend less time looking at the eyes, and this may represent, among many things, missed opportunities for social learning, especially if it happens very early in development. Could have in many ways kind of compounding effects. So here you see example eye tracking from an infant later diagnosed with autism and a control infant. And what you see is that the infants later diagnosed with autism spend more time kind of looking at the mouth here of this person and at this person's hands, whereas the infants who go on to be typically developing spend more of their time looking at the eyes and kind of the center of the face across the video scenes. And this is really interesting because it also fits well with what we're starting to understand about the genetics of visual attention. So there was a study that came out a few years ago that used a twin design. And so they did the same type of eye tracking paradigm showing social type videos to young and toddlers. And they did this in both twins that were fraternal, meaning that they share 50% of their DNA. They're not identical twins. And they did this in identical twins who share, roughly 100% of their DNA. And what they looked at was how similar are the viewing patterns across the videos between these two types of twin pairs based on the degree of genetic background they share. And what they ended up finding was that these monozygotic identical twins that share 100% of their DNA have strikingly different, similar ways of viewing these social scenes. And you can see that here. There's kind of a blue and a teal color that indicate where twin one and twin two are looking within the video. And they're really looking at very much the same aspects of what's going on in the video. Whereas if you look at the dizygotic twins who are in many ways similar to siblings, they share 50% of their DNA. They're looking at these scenes in different ways. They have some overlap perhaps in what they're looking at, but the sequence isn't so tightly locked, if you will. And so what this study says is, we know that autism is highly heritable, and we know that among typically developing people, how we view information in the world around us is strongly driven by genetics. And I think if you start to put all of this together with our work, looking at the infant brain and autism, you start to see how this developmental cascade might unfold. It may start with a genetic predisposition for autism within the family that ultimately, gives rise to differences in sort of the structure and function of the visual system. This may change the way in which a child views the world around them. And this change in what they take in from the environment may change the way also that the brain develops, because the brain is an experience dependent system. Whatever it's experiencing in the world around it alters sort of the saliency of that information to the brain. It reinforces certain types of connections in the brain and not others. So it may then have sort of a cascading effect on how the visual system relates to other aspects of the brain, and ultimately give rise to many of these sort of broad differences in brain development that we observe, and ultimately the core features of the autism phenotype in terms of clinical features. And so there's a lot of work that we have to do to refine. This is just a hypothesis, and we're still trying to kind of figure out how does this all work? What does it really mean? But I think it has really important implications for understanding how to intervene. Because what our work has told us so far is that differences in brain development precede differences in at least the core features of autism by a year or more. And if we want to intervene and have the maximal possible benefit for children with autism, we're probably going to need to do this before their first diagnosis. And I'm not saying that intervention at any time point in development is not important, because it is. But this work suggests to us that doing it early, during infancy, might really make a big difference in kind of what we see in terms of outcomes in children. And that, in many ways, is the goal of this IBIS early prediction project. So this project's goal is to think about MRI prediction as a way to lower the age of detection, or detection of risk, so that we can then intervene at the earliest possible time points in development. So the goal, that's sort of like the pipeline we think of for this project, is that, we go from general population prevalence of autism to infant siblings. So we've already narrowed down the prevalence in that infant sibling group to 1 in 5. But how do we identify which of those 1 in 5 are going to really need intervention, or may, actually go on to receive a diagnosis? And that's where we think potentially that an MRI in the first year of life may come in. We would then extract the relevant features of the brain, which we've been talking about before, and then we would build models that help us think about both the diagnostic outcome of the child, but also what are their specific areas of support that they may need. Are they more likely to have early motor delays? Are they more likely to have communication problems or joint attention problems? And kind of knowing that about each child may help us in the future tailor interventions. We would then evaluate, how well did these models work? And we would use various different combinations of kind of features to do this. And ultimately, the big picture goal of the work is to identify these infants that are at very high likelihood for autism, so that they could be assigned to early interventions or early intervention trials, so that we can kind of figure out, what's the evidence-based, what's the best evidence-based model for intervention for these kids. And this IBIS EP study involves a very similar overall structure to our earlier study, where every infant comes in for a visit at one of our 6 sites across the country at 6, 12, and 24 months of age. They receive MRIs and also behavioral assessments at these visits. For families, what this looks like for participation is it's a 2-day visit plus travel to the site, and this may be travel by plane or travel by car. The family usually comes and they stay overnight. We attempt an MRI scan with their child during natural sleep time, usually at bedtime. And then after each visit, the family receives an assessment on a report on how their child is developing. And many of our families find this to be a very useful feedback and tool, and many of them go on to use this to sort of gain access to early services as well. And I think the last thing I really want to impress about this study is that it is at its backbone an MRI study, but we have a strong interest and desire in making this a comprehensive study of how autism develops, but also what are people's support needs? Do people want early prediction information about whether their child will develop autism or not? What are the other aspects of child language learning or parent-infant dyad interactions that might support development? So we're doing various other projects kind of attached to this backbone of the MRI project. We're also doing EEG, which is a way to measure brain function in real time in infants while they're awake. And we're doing eye tracking as well to measure the same type of sort of visual and social behaviors. And we have a real interest in this new cohort and thinking about diverse perspectives in autism research. So I want to just highlight sort of 3 areas that this project is seeking to gain information and also provide information to the research and broader communities. So one of these areas of research is led by Kate McDuffie, and it's focused on the ethical, legal, and social implications of early prediction, right? So what do families want to know? Is it ethical to predict a later diagnosis of autism if early interventions are not yet in place or haven't been proven to be effective? So what does that mean? And we're trying to understand that piece of things. There was a webinar about this a couple of years ago. I'm sure if you Google this, you could probably find it still. I think it was recorded. We have another aspect of the study that's focused on identifying and addressing barriers to participation in research for minority families, because much of what has been done in the past has been focused on, sort of families that have the means to come and take part in studies. And we want to figure out how we can broaden our research to include all people on the spectrum. And then finally, we have a unique opportunity with the type of sampling strategy that we have to understand autism and autism features in girls and women. And in particular, thinking about sex differences across this developmental trajectory. And the reason is that we are not relying on traditional sort of methods for people to be detected for autism within the general community. We're watching these, in particular, young females develop over time, and we may be able to help us refine what is autism in females. So the broader goals of this study, are manifold. And the point at which we are now is that we are trying to complete our cohort. So we've currently enrolled about 180 families in this study. This study started right before COVID shut down, so we had a long pause, and now we're trying to kind of make up our time and bring in new families so that we can really do the work that we want to do. And we're looking to recruit an additional about 70 families into the study at this point. About a quarter of our sample so far has come to us through Spark Research Match. So that's been a huge success for this project. And I just wanted to say, if anybody on this call is interested in hearing more about our study, you can visit our website at the QR code here, or you can feel free to find me and email me about this. These slides will be archived after the fact, so you can kind of come back to this and look at our website again later if you'd like to. And with that, I will end, and I would be happy to take any questions. Thank you.
Thank you so much, Dr. Girault. This was so interesting and exciting to see this research happening. And I think what was really great is some of the questions that came in, your research is already answering. So they were answered later, and things like sex bias and diagnosing girls. But it's really exciting to see the progress. But that being said, we do still have some other questions, specifically about participating in the study itself. So for starters, obviously you're doing work with infant siblings, but is there any specifically with the older autistic siblings, to be eligible, would they need to have more profound autism, or can they be anywhere on the spectrum?
That's a great question. They can be anywhere on the spectrum as long as they've had a diagnosis of autism. They're eligible for participation. The family's eligible for participation in the study. We do screen out some genetic conditions. So if, for example, that you find out that your child has a particular genetic condition that's not heritable, therefore your younger child is unlikely to have that condition, that would be exclusionary for the study. But we would be more than happy to speak to any families about how this sort of operates, and we do talk to every family who's interested on the phone kind of about the study and how it works.
Great. And then with the children that are actually in the study, have you had any instances where they perhaps received a diagnosis while participating? And if so, what does that mean for the study? Would it disqualify them, or?
Yeah, so if the younger sibling enrolled in the study receives a diagnosis, that's totally normal. We expect that about 20% of the sample would, and they would definitely continue on with us. We provide at 2 years of age our own research diagnosis, provided by trained and licensed clinicians. So for many of the families in our study, that's the first time that their infant is receiving information from a clinician about an autism diagnosis, but it doesn't always happen that way, and that would be totally fine.
And then you mentioned earlier, about the importance of early intervention, or intervention at all stages. Are there specific types of therapies that you foresee helping with early brain development in these baby siblings that are likely to have autism?
Yeah, that's a really good question. So I think there are some sort of promising early intervention type models. I think of joint attention models being the ones that come to mind first. So maybe think of Jasper, if you've heard of that early intervention. That's targeted right now towards older children, toddlers. But I think that we could do something very similar when we think about how to train parents to sort of engage their children in joint behaviors early on. I think that's one type of behavioral intervention that we could see. Others will probably involve sort of sensory motor type stimulations, because we do see and by stimulation, I just mean sort of play. We do see those types of behaviors being really effective, for example, in like preterm infants, for helping them sort of get on a sort of more normative trajectory of brain development. So I think that there are some things that we could learn from that type of literature as well. But we're definitely still in the early stages of just learning about when to enter, like when's an ideal time to intervene, and learning, I think, a little bit about maybe that the visual system is involved will help us think about, well, what are the other ways that we do this? And I could go on about that a little bit more specifically, but I think right now we are thinking that there probably is sort of attention-based interventions that we imagine will be most effective.
And then in a similar vein, talking about early intervention, is there anything that would perhaps disqualify a family, perhaps early interventions or different therapies that are already being implemented in the home or with the siblings or,
For our study, no, not at all. I
okay.
mean, we ask about that type of information, and we try to keep track of it, mostly so that we can see if there is, a subset of kids who are all receiving early intervention starting at 18 months, do they end up looking different in terms of brain or behavior development from the kids in our study who didn't? But we're not really designed to test that, but it's in no way exclusionary.
And through this work and, through some of the findings and the different research that you talked about and the ongoing study, do you believe or have you seen clear themes in some of the traits or aspects of autism that are being, that are coming forth in through this research for kids? Have there been real true themes that appeared?
And you mean
Themes might be the wrong word, but just like common what like common traits of autism that you might be seeing in the siblings
yeah.
in their brain development? I guess that's more.
Yeah, so I think I'm going to answer what I think you're asking, and you can
Yeah.
correct me if that's not right. But so what we've started to see in terms of like how familial are some of these patterns of behavior, we see that language in particular is looking very similar between the older child in the family with autism and the younger child in the family with autism. And so we think, and these differences show up very early. So like by 1 year of age, infants, regardless actually of whether they receive a diagnosis or not, if their proband or older sibling had a language delay or has a language delay, that younger child is more likely to show that. And so we're thinking there are certain types of aspects, for example, of language development that might be a really important sort of early intervention target. But it's kind of difficult to think about how you intervene on language before children are talking, right? So we have to kind of think about what are the ingredients of language learning, and that might take us back to thinking about, well, what are the cognitive building blocks in the brain? And I think that's where we start to kind of get into some of these motor and sort of gestural communication abilities and how we might foster those things. So I think I kind of got at what you were generally asking, but feel free to rephrase if I missed
Yeah,
that.
I mean, hopefully that, answered the question for the person that submitted it.
Gotcha.
But I think even if not, it was a good clarifying point to
Yeah,
share.
sure.
Another question that just came in: how long will you continue to follow these younger siblings? And well, I guess I'll start with that first half of this question.
Yeah, so we're longitudinal study people at heart. So we feel that it's really important to think about development over time and how children change over time. In our original cohort, we followed those children from birth, and they're coming in now between the ages of 15 and 17 years old. So that was the original cohort. For this younger cohort, we have plans in the works to follow them at least through school age or school entry. Right now, we're thinking hopefully through the age of 6. And part of the reason for doing this is that we know that the sort of the cognitive demands of school can make sort of other types of behaviors come online that are not really appreciable in toddlerhood. We think of ADHD as one of these traits, right? So we often know, I mean, most children are not diagnosed with ADHD until kind of later in childhood or the preschool to school age transition. And obviously ADHD or attention type problems are very prevalent in autism. They're also very prevalent in high likelihood siblings who don't develop autism. So we're very interested in thinking about kind of how these trajectories unfold and how infant brain development might sort of predict or foreshadow a variety of later needs beyond just the sort of early intervention needs, but what might be school support needs that kids have later too.
Great. And that answered the second half of that person's question. So thank you for that. I think we have time for one more question, and this sort of gets back at the just actually participating, since you are still looking for participants. How can somebody participate if they perhaps don't live near a site? I know a center. I know you said there's travel involved, but what does that look like for someone who might be kind of far?
Yeah, so this happens all the time. So we basically have 6 catchment areas across the US that cover the entire continental US. I believe in the past, we've also had families participate from Alaska. So it's not out of the question if you don't live within the continental US. But what we generally do is we route that family to the closest of the research sites, unless there's a reason, like, for example, if the family, I don't know, lives in Texas, but they're about to move to New York City or something like that. But we generally route them to the closest research center for them and their family, and they choose whether they would like to drive or fly. If they're further than a reasonable driving distance and they don't want to drive, we are happy to do flights. And we often fly more than just one caregiver and the child, or we support the idea that many of these families like to travel all together. So we want to support our families to participate in whatever way makes sense for them. Some of our families, their probans are old enough that they're interested in doing a college tour by the time their sibling comes back for another visit later in development. So, in many ways, I think the families that have participated in the first wave of our study have seen this as sort of like a team that they're a part of and a science that they're a part of. And we now, especially as the children get older and are involved in the study, they really enjoy coming back and participating. So we're looking for families who are interested in the science and interested in participating, and we'll try to make that feasible for you kind of wherever you're coming from.